Chapter 14 explores the world of RNA. A recording is divided into 4 segments which will explore the role of RNA in gene function, in gene expression. We will touch upon the definition of gene. Afterwards, we will switch into discussing RNA structure and function, followed by RNA processing, splicing, and in the last recording we will touch upon the interaction between 2 or more RNA molecules. So we will start with question: what is a gene? When we discussed Mendelian inheritance in Chapter 3, we defined gene as an inherited factor which determines a characteristic. To study Mendelian transmissional genetics, this definition was appropriate because it enables us to define how gene influence the inheritance of traits. However, now we would like to go to more detail, to deeper level, and consider the physical nature of genes. So what is gene? In 1902, Garrett suggested that genes encode proteins. Gene was defined as a set of nucleotides that specifies the amino acid sequence of a protein. This is correct for many simple proteins. However, complication starts when you look for very complex proteins such as hemoglobin, which are built or made of subunit, and each subunit is composed of polyproteins encoded, or polypeptides, polypeptides encoded by different genes. So basically the definition one gene, one protein was revised, and it was suggested that genes encodes polypeptides. One gene, one polypeptide. Now, when Francis Crick in 1958 proposed his theory about genes and protein, he suggested that genes are collinear. What does it mean? It means that continuous sequence of nucleotides in DNA encodes for continuous sequence of amino acid in the protein with intermediate sequence in RNA. Now, this collinearity is still applied to the inheritance to genes in bacteria and many viruses. However, the situation is much more complex and complicated in eukaryotes. So what about eukaryotic cells? In eukaryotic cells, gene structure is fundamentally different, and genes contain more DNA than what is required to encode proteins. In addition, early experimentation suggested that in nucleus there are large, long RNAs which we do not see in cytoplasm. And when the experiments were performed, as one outlined right here, where you take DNA, you extract DNA, and you extract RNA from the same cell. Now you denature DNA, so you create single-stranded DNA, and you incubate it with RNA. And you know, when you have complementary sequences, they would hybridize and anneal. So what you will see after hybridization double-stranded DNA, and also some DNA annealed with complementary RNA. However, you will see these so-called bubbles where actually sequences, so-called non-coding region, have form loops. If you do the same experiment in bacteria, you will see DNA and RNA align equally. So this was first suggestion that non-coding regions interrupt coding sequences within eukaryotic genes. So based on previous experimentations, respective experimentation described in previous slides, it has been suggested that eukaryotic genes may possess coding and non-coding sequences. Coding sequences are called exons and non-coding sequencing introns. Average human gene might have anywhere from 8 to 9 introns. However, it may vary. There are genes which have very few introns and there are genes which have many introns. All classes of eukaryotic genes encoding ribosomal RNA, transfer RNA, mRNA may contain introns. And size and number of introns may actually defines the complexity or go along with increased complexity of the organisms. There are four major types of introns, and they are outlined right here in this table. Group 1 and Group 2 are so-called ribozymes because they can self-splice. Group 1 introns which self-spliced you can find in eubacteria, in bacteriophages, and some eukaryotic genes. Group 2 self-splicing introns are found also in bacteria, archaea, and eukaryotic organelles such as mitochondria and chloroplasts. There are several differences between Group 1 and Group 2 complexity of secondary structure. However, one of the major one is that Group 2 when splicing creates a lariat structures. Now, the major group of introns which we will focus on and spend more time discussing are nuclear pre-mRNA introns. Those are introns which you can find in protein coding genes in the nucleus of eukaryotic cells. To splice these introns, a cell uses spliceosomes. Spliceosomes are huge complexes which comprises proteins and small nuclear RNAs. And the last group of introns we can find in tRNA, in eubacteria, archaea, and eukaryotes, and this can splice enzymatically. So now, after we discuss complexity of eukaryotic genes, which are composed of exons and introns, we have to revisit the concept of the gene. So what is gene? If we stick with the definition that one gene is one polypeptide, that the gene encodes for polypeptide sequences, so we could consider as a gene only exons. However, because we know now that the structure of genes is much more complex, the concept of gene was changed, and it was proposed that we define gene as a DNA sequence that codes for all exons, introns, and sequences at the beginning and end of the RNA that are not translated into protein. So what are those 5' and 3' sequences, so-called 5' UTR and 3' UTR? Untranslated regions. Even though these regions are not translated into proteins, they are critical for RNA stability and for translation of RNA into protein. Now, some geneticists went even further and proposed that we define a gene as the entire transcription unit. You remember we discussed transcription unit in previous chapter. So now when we discuss what gene is, we know that actually genes encodes for polypeptides, and one gene includes all exon, introns, and 5' and 3' UTR. So let's talk now about mRNAs. So what are mRNAs? Basically, mRNAs are short-lived RNAs which carry information, genetic information from DNA to proteins. If you look in bacteria, mRNA is transcribed directly from DNA. And you know, bacteria do not have nuclear membranes, so as transcription is going on, translation starts. So translation starts before transcription is completed. However, in eukaryotes, cells, the situation is more complex because the nucleus is separated from cytoplasm by nuclear membrane. So in eukaryotic cells, DNA is first transcribed into so-called primary transcript pre-mRNA, which still contains sequences of all introns. This pre-mRNA is afterwards processed, modified on 5' and 3' ends, and spliced into mature mRNA. So pre-mRNA goes to mRNA, and basically only after this processing, RNA can be transported into cytoplasm and serve as a template for protein synthesis. Now here in this picture you have actually outlines the structure of mature bacterial mRNA. So bacterial mRNA has within this 5', this is that 5' UTR, and here is 3' UTR, those untranslated region you find in mRNA, also in eukaryotic. In bacteria, this 5' UTR contains a sequence which is upstream of start codon, which we call Shindal-Garno sequence. You can find it only in prokaryotes. This sequence is necessary for recognition of ribosomes. The sequence in Shindal-Garno region is complementary to ribosomal RNAs and basically is important for association of mRNA with ribosomes. Afterwards, you have the protein coding region, which is defined by start codon and stop codon. And after stop codon, there is 3' untranslated region. Pre-mRNA and processing of pre-mRNA into mRNA in eukaryote we will discuss in more details in the next segment.